Assalamu alaikum. Bismillahirrahmanirrahim. Alhamdulillah. Alhamdulillah wa kafa wa salat wa salam ala ibadihi allazina istafa amma ba'd. Faqala Rasulullah sallallahu alayhi wa sallam ma anzala Allah min da'in illa anzala lahu shifan. Almighty Allah has not sent down any disease except that He has also sent down its cure. Rabbi shrah li sadri wa yassir li amri wahlul 'uqdatam min lisani yafqahu qawli. Good afternoon, Dr. Fahmi, Dr. Meena, distinguished guests, colleagues, ladies and gentlemen, very good afternoon. As you know, the topic assigned to me is the traditional complementary and alternative medicine with a special reference to herbal medicines, their quality, safety and efficacy. Well, ever since the incorporation of traditional medicine in its program, the World Health Organization has gone a long way. In 1976, in its program in 1976, the World Health Organization has gone a long way in creating a worldwide interest for the development and promotion of different systems of traditional medicine and their best utilization in the healthcare facilities. Well, it is being categorized in two as medication and non-medication. Medication as medicinal plants, mineral and animal materials. Non-medication, physical, mental and spiritual therapies, manual therapies, osteotherapy, chiropractic, acupuncture. It's my pleasure to say, really, I appreciate that the organizers of the committee has put all the systems, they have distributed one of the categories to all the speakers. Since morning we are listening chiropractic, naturopathy, traditional Chinese medicine, and I think Ayurveda, homeopathy, acupuncture, cupping, Unani medicine just now, ozone therapy. Well, my part is the herbal medicine. Recently, the National Center for Complementary and Alternative Medicine, NCAAM of the National Institute of Health America, have announced the funding of 3 new centers for the study of traditional herbal medicines and other forms of complementary alternative medicines. Well, international health research is one of the 8 areas of explained in their strategic plans. Well, according to the global industry analyst, the global alternative medicine sector is expected to reach close to $115 billion by the year 2015. And this market growth is fueled by a trend towards herbal and nature-based products based on the presumption these products causes fewer side effects than the modern medicines. With due apologies. Well, my part is the herbal medicine, and according to World Health Organization, the plant-derived material or preparations with therapeutic or other human health benefits which contain either raw or processed ingredients from one or more plants. In some traditions, material of inorganic or animal origin may also be present. Well, herbs or plants have always been a common source of medicament, either in the form of traditional preparations or pure active principles. In a survey done by World Health Organization, it has been estimated that 80% of more than 7,000 million, that is the 7 billion inhabitants of the world, rely mainly on traditional medicine for their primary healthcare needs. And it can safely be presumed that a major part of traditional therapy involves the use of plant extract or their active principles. Well, in developed countries too, plant-derived drugs are considered important. For example, in the United States of America, 25% of all the prescriptions dispensed from community pharmacies contain plant extracts or their active principles. Of the conservatively estimated 300 to 400,000 species of the higher plants that exist on Earth, almost 10 to 20%, that is almost 35,000 to 70,000 have been employed in traditional medicine. Now, the efficacy of these drugs is due to the presence of one or more biologically active chemical constituents. But unfortunately, quite a number of these drugs are frequently adulterated with cheap, less potent, and spurious plant materials, either posing danger to the public health or they are not so efficacious as expected. Why? Because the therapeutic efficacy of these drugs will depend on the purity and economic availability. Well, in the recent year, with ever-growing commercialization in the field of herbal medicine, there has been an instant demand for the quality control of the drugs used in this system. And for this, standardization is usually recommended as a solution to the problem. The studies on the identity, purity, and quality of the genuine drugs will enhance information in checking the adulteration. A set of standards would no doubt be deterrent in checking the adulteration. Rather, it will be an aid to the drug law enforcement. But what is conveniently overlooked, that the standardization of the materia medica of herbal medicine itself is a difficult task. And therefore, before dealing with this topic, let us have an idea or let us recapitulate what is meant by standardization. The standardization may be defined as elimination of unnecessary variety, or in other words, it implies the collection of the relevant data, putting it down in a logical sequence, eliminating the varieties and inconsistencies before formulating the standard. In order to appreciate the problem facing herbal drug standardization, let us examine the situation in totality. Well, the term totality stands for examining the problem of standardization from the time of plantation of a drug to the time it is dispensed to the patient, either in the form of crude products or in the form of finished product, capsulated form. The word standardization includes various problems and fields of study. The standardization includes study from birth of the plant to its clinical application. Obviously, the scientists are looking after isolated parameters, but one should have a detailed idea of the combined approach. The study of the plant includes its cultivation, source and period of collection, identification, storage, chemical composition, preclinical studies to its clinical approach. Let us see what are the major causes for the inconsistency of these herbal drugs, and that are regional or the linguistic nomenclatures, eco-chemo or phenotype variations, harvesting period, method of drying, storage conditions, mode of grinding, and the particle size, extraction procedures, nature of formulation, as well as the shelf life period. I will deal with all these topics. Let us start from the very beginning, that is the good agricultural practices. Any medicinal plant to be cultivated should be recorded by the scientific name, that is the genus, species, subspecies, variety, cultivar name, eco, chemo, phenotype, authoritative name, family name, and if possible the local and the common English names. Besides having the documentation on the medicinal plant itself, the growers and producers shall prepare the botanical voucher specimen for submission to the regional or national herbaria. And these voucher specimens shall be retained for a sufficient period of time and should be preserved under proper conditions. The seeds and the other propagation material should be verified botanically. Their origin and breeding history should be checked, and the providers of these materials should ensure the quality and the identity and the quality of these materials. Cultivation. As you know, that different plants have different habitat. The habitat of the herb will ensure the uniformity of activity in relation to the source. The very first step should therefore be the cultivation of the medicinal plants under controlled conditions. The prerequisite for this is the collection of authentic specimen of such plants from herbal system of medicine and the subsequent investigation of method for large scale cultivation. Cultivation of medicinal plant requires intensive care and management. Site selection. Medicinal plant material derived from the same species may have a very, very different properties if they are being at different sites, if they are being cultivated at different sites, one in Abu Dhabi and the second suppose in Oman. So basically this is very important parameter. Ecological environment, that the cultivation of medicinal plant may affect the ecological parameters of the flora and fauna of the environment and vice versa. The soil should contain sufficient amount of nitrogen, sulfur, phosphorus and other elements in order to have optimal plant growth. Irrigation and drainage should be controlled and should be carried out in accordance to the need of the individual medicinal plant species. The plant and any chemical used in the growth and protection of the medicinal plant should be made to a minimum amount if there is no alternative method. Otherwise, some pruning, tanning and other things should be used. Medicinal plant should be harvested in a proper season or time period in order to have the production of the best possible source of best possible you can say the crude plant material and the subsequent finished product. The time and medicinal plant, the time period for the harvesting should be selected on the basis of the qualitative and quantitative estimation of the active constituents, active principles, rather than the total vegetative yield of the crop. The personnel should have a high degree of hygiene is required from the side of the personnel, and moreover those who are involved in this plantation, they should know the soil, pH and the plant they are going, so it's important. Good field collection practices. Basically a technical planning is required for that. That is the population density, the topographical map, geographical distribution for the target medicinal plant. Of course, the prior permission for the collection of medicinal plants should be obtained, and only those medicinal plants should be collected which has been reported in the regional or the national pharmacopoeia. Ecologically non-destructive means of collection should be adopted. So we will deal one by one. These are the intellectual property rights and the threatened and endangered species. These are the two ethical and legal issues while we deal with the agricultural as well as with the field collection practices. These are the segments for the post-harvest care of the medicinal plant, starting from collection, harvesting, drying, gardening, packaging and storage. Different parts of the plants, there is different timing and seasons to get it collected. That should be taken care of. Drying of medicinal plants is very important parameter, and what particular temperature, probably it is 60 degrees centigrade and written wrongly with 600, 30 to 60. And it should not be dried in the sun, it should be dried in the normal shade. So it is an important protocol. Well, proper storage and preservation method of the plant materials are essential for maintaining a high degree of the quality of the drug. Some plants, they are particularly liable to deterioration if they become damp during shipment or storage, for example, cascara leaves. Well, medicinal plant, if not well packed, they absorb moisture, it triggers enzymatic activity leading to the spoilage and storage. As aware, the organic constituents of the plant have a limited shelf life, and these compounds undergo degradation under the influence of heat, light and moisture, particularly when standard conditions for the drug collection, drying and storage are not maintained. Attack of pests is mainly seen while keeping the plant material before mixing it with the other ingredients. The moist and the sunny situation might cause hydrolysis to occur. The chemical constituents will be changed and the efficacy of the drug will be, of course, deteriorated, changed. And that is why the storehouse should be inaccessible to the rodents, pests and insects. It should be cool, dark and well ventilated with the dry air. These factors cause partial or sometimes complete loss of the thermolabile active principle. And these variations are too diverse for the standards to be laid down. Well, above all, the identification of the single drug is an important parameter. And nowadays, due to the adulteration, the minute differences of the medicinal and non-medicinal plants should be distinguished. Misidentification of a Chinese plant in a slimming preparation is believed to be responsible for serious toxicity, renal, intestinal fibrosis, experienced by some 70 Belgian women. 30 of them, they needed dialysis or kidney transplantation. And that was due to, between these two plants, Stephania tetrandra and Astrologia fangchai, and was thought to have been arisen due to similarities between these two plants, as you can observe. As aware, astrologic acid are well known for its nephrotoxicity in human as well as in certain animal species. They are potent carcinogens in rodents also, and that is why restrictive regulation has put forward in the year 1981 in West Germany for the preparations that contains astrologic acid. Let's take a normal example of Crocus sativus, everybody knows it. Saffron, that is the dried style and stigma of Crocus sativus, contains 3 crystalline metals, alpha, beta and gamma crystalline, having 0.7 to 0.3% of saffron. Besides having refrigerant, tonic, diuretic and stimulant properties, saffron has anti-inflammatory, antiseptic as well as aphrodisiac properties, for which it is being incorporated in many of the single as well as the composite preparations. Since it is a costly ingredient, the adulteration is on its peak. To differentiate between the adulterated and the pure, the structure of the stigma, which is slightly expanded in the form of a narrow tubular structure and plays an important role even with the hand lens. Sometimes identity by ordinary taxonomic means is a difficult job and it requires various other fields of study. Take the example of a plant which has a controversial identity is being sold in the market of different countries in form of isolated underground pot with some remains of the radical leaves. The botanical identity of this medicinal plant is highly controversial because some 15 species belonging to 10 genera and 3 families, they are being sold with the same trade name. It is called as Ratanjot in India and Pakistan, Yadlong in Afghanistan, and Abu Khalsa or Saleh Hamam in Arab countries. These are all vernacular names. The chemotaxonomic and the phytochemical and chemotaxonomical studies pharmacognostic for the boryginaceous plants lead us to a conclusion that the plant Ornibia nobilis is the required or the genuine plant material. Pharmacognosy remains the best means for identification of a drug, especially that of the plant origin. Where the drug used is a whole plant or its part, it's fairly easy to get it identified botanically. But when for an unorganized drug or when the drug is reduced to powder form, a greater degree of expertise is needed on the part of the pharmacognosist. Moreover, pharmacognosy by itself doesn't provide all the information. And standardization doesn't stop with the identification, but extends to quality and potency. Recourse to the physical and chemical method of standardization becomes necessary, or let me say rather a must. Chemical standardization is possible if we know the limits of variability of any characteristic constituent of the drug present in a measurable quantity. The compounds present in a small quantity may not be useful for standardization purpose. And the isolation of a single compound, as you know, is a difficult task. And to press them into service for standardization purpose is even more challenging. These chemical studies, the standardization of herbal drugs on the basis of chemical studies has got its own advantages, exactness of results, quick and less time-consuming observation, and recovery of the material in the cases where the drug supplied is in small amounts. Although there are several methods for the standardization of drugs chemically, however, it depends upon the nature of the drug under study as to what particular method should be adopted. No single method can be listed for it, but however, there are certain common parameters which can be established. Well, in the case of the whole drugs, the macroscopic and the sensory characters are usually sufficient to enable the drug to be identified. The general appearance of the sample will often indicate whether it's likely to comply with such protocols like that of percentage of seeds in the case of colocynth, ash in valerian, and alcohol insoluble matter in the case of Asafoetida heathii. Well, the price of certain drugs will depend mostly on the size and color, which are not necessarily related to the therapeutic value, and this implies to some very important drugs like that of senna, chamomile, and rhubarb. There are some specific quantitative parameters which have to be performed in certain specific drugs as well as some families, for example in stevia, sweetening index, and so on and so forth. Both the organic and inorganic constituents have to be identified. The identity and purity of the drug, for the identity and purity, the foreign organic matter, pesticide residue, radioactive contamination, and heavy metals, these has to be performed, and they have their own importance. Let me talk about the heavy metal. It should be in the prescribed limit as given by World Health Organization or FDA. And the importance of this, you can understand, might be you have read and used that forensic studies of the China's second to last emperor, some 100 years ago, Guangzhou, he died of arsenic poisoning. And the tests, the forensic test revealed from his body hair, it was 2,000 times higher than the normal or the ordinary people. And the comment of the experts that Guangzhou died of arsenic poisoning. So this is very important to get, to carry out all these instruments, all these parameters. Microbial contamination should be taken care of, and it should be within prescribed limit as per WHO organization. A crude drug may be assayed for a particular group of compounds. For example, total alkaloid in belladonna, total glycoside in digitalis, and alternatively reserpine contained in the case of Rolfia serpentana, which is slightly different from the other alkaloidal content. These all parameters should be performed. The successive extraction should be performed in order of polarity, increasing polarity of the solvent from petrol to alcohol. By the use of thin layer chromatographic studies, it's easy to find out the number of the constituents present in various extracts of a drug. Well, the retention time as well as the RF value of a compound determined under a specific condition is characteristic, and that can be used as a standard for that particular compound. The fragmentation pattern and the molecular weight will further facilitate the identity of the compound. Let us take a normal example, echinacea syrup, which is normally available for the common cold and cough. Suppose we have to standardize this drug, we will see that whether it is the Echinacea purpurea or some other varieties of Echinacea like that of Palisida, Angustifolia, or so on. So chicoic acid is the marker compound. So we have to find out the marker compound. And what is the percentage of chicoic acid? On that basis we can say that it is Echinacea purpurea. Suppose Ami visnaga is admixed with Ami majors is admixed with the Ami visnaga. Khilla baldia and khilla shaitani. So khilla shaitani is, suppose it is mixed with the khilla baldia. So how we can identify? We just take the transverse section of the pericarp and the presence of the four prominent secondary ridges as well as the absence of lacunae on the outer vascular bundles. It will be a good indication of the exact plant. Moreover, Amimatus contains 5-methoxy and 8-methoxysoralin, whereas the visnagine contains, the Amivisnaga contains visnagine, besides the common factors of furanocoumarine. And that can easily be identified either using thin layer chromatographic studies or high performance liquid chromatographic studies. Normally, the separation and isolation of the chemical constituents, we use planar chromatography for that. Well, just to make it in a short trick, the standardized extract, what are the parameters to be taken care of? To start with the plant material, the nature, size, origin, degree of processing, moisture. In the case of solvent, the nature, concentration and polarity of the solvent, whereas for the manufacturing equipment, the filling height, hydrostatic pressure, batch size. But for manufacturing method, these are the important parameters like type, time, flow velocity, temperature and pressure. All these things should be maintained for having a standardized extract. So basically, when all these critical parameters are maintained, an extract can be called as the validated extract or a standard extract, and it can be referred in future. Basically, the instrument which we use is the Bucci rotavator as well as the freeze dryer machine for making it into dosage format. Current good manufacturing practices, as you know, is a method for analyzing that the products are consistently produced and controlled according to the quality standard. It is designed to minimize the risks involved in any phytopharmaceutical preparation that cannot be eliminated through testing the final product. These are the specification of the finished herbal medicine product in different dosage format. Well, in a nutshell, what I have described up till now, we can say it is a good medicinal plant practice, GMPP, which starts from the good agricultural practices, good field collection practices, good horticulture practices, good laboratory practices, and of course, coming to the good manufacturing practices, and after that, the good marketing practices, and then and only then we will be able to get phytocosmetics, functional foods, OTC drugs, or the package of the drugs. So all these things should be taken care of.
5 minutes.
Sure. I'll try to simplify it as possible. The stability studies has to be performed in order to say, before it was a very long process, but due to accelerated studies, maintaining the temperature, pressure, and all these things, now it can be in a very short time. Yes, for the quality of a licensed herbal medicine, it is not only essential to have identification of the plant itself or its species, but it is equally necessary to have a batch-to-batch study. All the batches, either collected from Abu Dhabi or from Sharjah, from Ireland, it should have the same chemical constituent. Pharmacovigilance, as you know, it strengthens the credibility of the herbal drugs. A very important protocol nowadays is the adulteration. Now, unfortunately, quite a number of the licensed herbal medicine have been found to be adulterated with the non-prescription and even prescription drugs like that of phosphodiesterase 5 inhibitors, sildenafil citrate, Viagra, tadalafil, vardenafil, or their analogues. The imperceptible use of these analogues is very dangerous, as they have not been tested formally for their safety and efficacy. And for the public health, in view of the potential harm to the public health, there should be a very proactive measures should be taken against these pharmaceutical analogues and to lift the guard against for the public and the consumers. Well, the adulteration is found in the herbal analgesic drugs like paracetamol. The conventional drugs are being added. Similarly, nowadays dietary supplements are there. Obesity is a very common in UAE also. So a number of the conventional drugs like orlistate, acomplia, dithylpropion, they are added. And not only this, but the drugs which are banned by the Ministry of Health, by Health Authority, by United States, by FDA, like sibutramine, hydrochloride, phenolphthalein, they are banned through and through. They are being added. Yesterday I was doing an experiment and found 2 consecutive conventional drugs being added in a sibutramine, and they put it as the name of Sabri. However, for the cholesterol lowering herbal drugs, a number of these statins are being found, and the latest format of these statins, cortisone and corticoids are being added. For example, somebody wanted to reduce and somebody wanted to gain weight, cortisone is being added, or corticosteroids are being added. Antihistamine drugs, cyclopentadiene, are being found in many of the herbal drugs. Well, analgesic herbal drugs are being found adulterated with the sulfonyl areas like that of the gilbertlamide, tolbutamide, glucophage, and the latest format of is acetohexamide, tolazamide, and many others. The abnormal use of these sulfonyl ureas and their abnormal doses, it may cause a profound hypoglycemia. This is how we check this. So basically, even if the efficacy of a drug is recognized and traditionally accepted, it must be tested it must be guaranteed for the quality and safety. Since quality is the paramount parameter because it affects the efficacy and safety of the drug being used. So nowadays, we see that the product quality ranges from very high to very low due to three main factors: intrinsic, extrinsic, as well as the regulatory factors. So this standardization is a challenge for the scientists due to the volume of the work, absence of research data, complexity, lack of initiative in manufacturing, flooding propriety, and many other reasons. However, let me conclude the things by 2-3 last slides. The reproducible efficacy and reproducible safety will depend on the reproducible quality, and therefore any herbal drug, or let me say, because before me there was a Unani lecture and after me there will be a Ayurvedic lecture. So any of the complementary medicines, any of the alternative medicines which want to be regarded as rational drug, it should be, it need to be standardized, and it must have a quality. Its quality must be tested. So basically the herbal drug resurgence, I'm going in very fast, I'm not going to read it, worldwide, basically this 15% increase as compared to the modern drug, which is 3% only. In United States for the last 2 years, 50% during last 2 years the products are common. China, USA, Japan, Malaysia, everywhere you can see that there is and I will show you in the graph, the global market for the herbal products of cardiovascular diseases 27.2%, respiratory disorders 15%, digestive and tonics 14.4%, for miscellaneous disorders 12%, and similarly sedatives 9.3%, and so on and so forth. Let us see from 1995 what is the trend of the herbal sale in the global market. Let us see in the United States, it starts, you can see, it's always in an increasing order. If you see in 2008, it was US dollar. In 2008, it was $60 billion, the world market of the botanicals. And now, if we see in the last decade from 2000 to 2010, we can see it, and you can observe it, there is a consistent and continual increase in the sale of the herbal drugs in the last decade. And not only for the single drugs, but also for the compound drugs, which is being mixture of more than one. And the same ratio you can see. Well, this is the concluding things which I wanted to say. As you know that standardization is a dynamic process, and today's standard will become obsolete tomorrow. Obviously, there is a lot of work has been carried out, not only here, but globally. But still, there are certain dark recesses which have to be eliminated using the high throughput technology, using the random amplified polymorphic DNA fingerprinting, hyphenated NMR techniques. These genomics, these are the instruments which are being used for the identification of compounds over there. And the last slide, which shows that these genomics and metabolomics, it seems to be the future of the medicinal plants, herbal medicines, and herbal medicinal products. Actually, I have cut short many things because I have been warned by in the last but not of least important, I once again want to thanks to the organizer for inviting me, for giving me a chance to present my views against this August gathering, to the Health Authority Abu Dhabi, and also to the Director PHR for giving me a chance to present this paper over here. Thank you once again. And also for the audience, you have a very patient listening. Thank you once again.
One or two questions, you can take one or two questions.
Really, I was in India 2 months. I delivered paper.
Congratulations.
What I told the people, even I tell all the people here, the challenge in our area, it is with the quality and safety. This is the real challenge to deal with the herbal products for the consumer. It is really big challenge in our area, the quality and safety. The law and regulation, what he mentioned, good manufacture practice, We say in the food, the same in the herbal. We should take care for the herbal and the food from farm to mouth. This is really, we should take high concentration about packaging, about storage, not only storage, even from the storage to the products reach, herbal products reach the consumer, the distributor, the way of the distribute things. In my own eyes, we see in our area, either in India or in China or here, or most of the Arab world, our area, the herbal products, really, the American and the Canadian and the European, unfortunately, they take the active ingredients from our area, they bring it back to us with high price because of the quality. Why? Because we do not fulfill the challenge of the really produce the herbal products from farm to mouth. As he mentioned, this is very, very important issue for anyone who would like to enter this market. The market, as he mentioned, is huge. You are talking about maybe $200 billion now, the market of the herbal. Thank you very much.
Thank you very much for your comments, and I appreciate your observations. And actually, this is the correct situation. Last month I was in USA and
because I used to be chairman of the consumer product.
That's what I'm telling.
I feel this very, very good issue for the consumer.
No, that's why I am thanking you for your observations, and this is the real fact. As I was telling, last month I was in Turkey as well as in USA, and in the coming month they have invited me to chair a session in the United States, and the position over there is exactly the same. The herbal medicines are growing rapidly, continual and consistent increase in the herbal sale. So it's a very important field. Thank you very much for your experience.
Thank you, Dr. Muhammad Kamil.